Hello everyone, my name is Anthony Edward Szego. I work at Stockholm University. Today's talk within the Green Carbon webinar series, I will talk about heterogeneous catalysis applied for environmental applications. So first, I'll give a brief overview of what I'll discuss in the talk. I'll give a brief introduction into what catalysis is and why it's useful for us. And then I will go into two main project topics that I apply catalysis in for environmental applications. And lastly, I'll give some conclusions of what I've presented. So as an introduction, we first have to ask ourselves, what is a catalyst? So a catalyst is a substance that modifies the rate at which a reaction takes place without itself being consumed in the reaction. The way a catalyst does this is by, as we can see here in this image, this is a reaction coordinate versus energy plot. So we see the reactants at a level and the products at another level. And then without a catalyst, the reaction would go through this purple pathway, which has a high activation energy. And as we know from the Arrhenius equation, the rate of a reaction is influenced by the activation energy. The higher the activation energy, the slower a reaction, the slower the rate of a reaction is. So when we add a catalyst to the system, what we do is find an alternative pathway for this reaction to take place. And this reduces the activation energy for the reaction, making it go faster. But it does not alter the thermodynamics of the reaction. So the thermodynamics of the reactant and the products are all stay the same. It doesn't modify that. If not, it wouldn't be a catalyst. So with this in mind, we can say that we can categorize catalysis in three main groups. There's homogeneous catalysis, in which the reactants and the catalyst are all in the same phase. This could be gas-gas, which is less usual. The most usual one is liquid-liquid or solid-solid. Then there's also heterogeneous catalysis, in which the reactants and the catalyst are in separate phases. So this could be liquid gas, or solid liquid. And then there's also biocatalysis, which is the catalysis that takes place within biological systems. And in this case, the catalysts are called enzymes, which you might know from different classes you may have taken or whatever. And why are catalysts important? Well, they're important because they make reactions possible, which wouldn't otherwise be viable. So for example, the reaction of hydrogen with oxygen to give out water. It's thermodynamically feasible, but the activation energy of activating the hydrogen molecules and the oxygen molecules is very high. So without the use of this catalyst that would reduce the activation energy, the reaction wouldn't take place naturally. Another reason is that using catalysis leads to high yields and product purity within the reaction. So we can enhance the yield of a reaction, and the catalyst can be selective. So if we have a reaction that can give different products, by using a catalyst, we can selectively get one of the many products, which would then be useful for when we need extra pure components or something. We don't have to go through a step of separating them if we manage to make a catalyst selective to one of the products. And lastly, the use of catalysts reduces the amount of reactants needed and the side products obtained because of this selectivity and this high yield. We get a lot of product with an X amount of reactants. And since if it's selective, we don't get side products. So this is catalysis is fundamental in this green chemistry that we're trying to achieve. So in my work, what I do is prepare materials that I use as heterogeneous catalysts, and I apply them into different reactions. One of them is the carbon dioxide to methanol reaction, in which we transform CO2 with a reaction of hydrogen and produce methanol. So it's a hydrogenation reaction. And a second set of reactions, which is the biodiesel production, which goes through a transesterification reaction that I'll show you after. So entering into the catalysis that I do, the first one is CO2 to methanol. As we can see the reaction in the top of the slide, it's fundamentally CO2 is hydrogenated with hydrogen and it gives out methanol. Why would we want to do this? Well, as we know, CO2 is a greenhouse gas. So increase of concentration of these gases in the atmosphere is what's leading to global warming. So if we could find ways of sequestering and using this CO2 to make something useful instead of letting it go into the atmosphere, we're winning, basically. So if we would be able to obtain hydrogen through renewable sources like solar power energy or wind energy, and we sequester the CO2 that is used up in industry, we can react both of them and we get methanol, hopefully. It's our target. So methanol is a very useful molecule because it's used in many different applications. So we could make polymers with methanol, which would be green polymers because they're made from renewable sources. Methanol is also used as a solvent in many applications, pharmacological industry, organic synthesis, plastic production, everything. Methanol is quite used as a solvent. We can also use methanol as a fuel by itself as methanol, or we could use methanol in transesterification reactions to produce biodiesel that I will explain afterwards. And then when we use up this methanol that we produced, it goes again into CO2. So if we find a way to recapture that CO2, and then we can make a whole cycle of methanol efficiency, which is called the methanol economy, basically. This is methanol economy in a nutshell. So what I do is this transformation of CO2 to methanol. We based this project on a homogeneous system. So this homogeneous system has two phases. It has an aqueous phase that contains amines dissolved in it, and then there's an organic phase that contains the a homogeneous ruthenium catalyst. So as I said before, homogeneous means that the catalyst is in the same reactant. In this case would be liquid-liquid. And the function of the aqueous solution is to sequester the CO2. So aminated solutions is a very well-known process to capture CO2. And then if we combine this capturing with this homogeneous catalyst, we obtain the synthesis of methanol through hydrogenation reactions. So what we're trying to do is heterogenize this system. So make it a heterogeneous system in which the catalyst would be in a different phase from the CO2. In our case, it would be a solid phase and the CO2 is in a gas phase. So how we're doing this is through modification of silica. So we start from commercially available mesoporous silica particles. Through a grafting process, we make this layer of polymer on the particles of the silica. So the role of this aminated layer of polymer is the capturing of CO2. This is all in one phase, so this would be a solid phase. And then we further functionalize this system by impregnating ruthenium catalyst. So the same one they used in the homogeneous system, we're just anchoring these active sites on this material. So we get a bifunctional material that would allow the capturing of CO2 through this aminated polymer layer, and then the transformation of this captured CO2 into methanol through a hydrogenation reaction. So this grafting process, the general reaction is shown down here in where it says grafting process. So this side on the left would be the general surface of silica. So it's got these silanol groups that can then react with these monomers that would be the aminated monomers. So this R chain varies in different lengths and different types of amine groups. So we've tested out 3 different types of amine groups. We have a primary amine, a secondary, and then a monomer that has both the primary and the secondary. impregnations or on itself has a capacity to absorb CO2, but it's mostly fissile CO2. And for these transformations, we prefer to have chemically adsorbed CO2. So by the modification with these aminated monomers, we see an increase in the capacity of CO2 capture. So all in all, we can say that modifying silica with these aminated silane monomers leads to enhancement in the CO2 sequestration. And then, as I said before, we further functionalize these materials by impregnating a ruthenium homogeneous catalyst, which we can see here. It's got a rather complex structure. It's a coordinated complex. So the impregnation we do is just, first we dissolve this homogeneous catalyst into an organic solvent, and then we add this amine modified silica, and we basically just let the solvent evaporate, so all the catalyst is anchored on the surface. We don't know exactly yet how these ruthenium active sites are anchored on our material. It's probably through these, as you can see, these complexes have a ligand that has an amine group, so they're probably displaced by these monomers before in the aminated polymer. But within our research group, we have a person in charge of doing some computational studies on the structure and stability of these materials. So we test the energy, the theoretical energy and stability of different forms that this catalyst can have on the surface. And we find the lowest energy one, and that would be the most stable. So with the preparation of the material, we take this material into a reactor, and it's a batch reaction that we do. So first we load the reactor with our prepared catalyst, and we saturate the reactor with 2 bars of CO2 for 2 hours. This would saturate, as I said, the material, and it would absorb as much CO2 as it can. And then after this saturation is finished, we insert hydrogen into the system, and we heat it up to 155 degrees for 40 hours. So we get as much conversion as possible. And in this time, the ruthenium active sites will allow the hydrogenation of this captured CO2 with the hydrogen that we insert into methanol. So after the reaction is finished, we extract with using a vacuum pump as a suction. We extract from the reactor the methanol that is formed, and to quantify and see if we actually did form any methanol, we're using liquid state proton NMR. So in this image, we can see an example of proton NMR spectrum. So we have the blue line is a loaded reactor without this ruthenium modification. So it would be just the aminated silica. And in red is the result of the reaction with this ruthenium modification. So as we can see, the area of the peak allows us to determine the concentration of methanol in the system. So we see that the system works. So we are indeed generating methanol through catalytic activity of the system. Another study we did is going more into the fundamentals of the material. So we used solid state NMR on the modified silica without the ruthenium to try and understand how the CO2 is adsorbed into the material. So we found in this image, we can see two spectra. The red one is a dry sample, so it's been handled in a glove box and sealed for it not to have any contact with air or any water. But we can see that there's, well, I'll explain that later. And the blue one is a wet sample. So we handled these materials within a water atmosphere. So what we see in this sharp peak here that we see in the wet sample that we do not see in the dry sample is the existence of what we think is a bicarbonate species. So this is a currently open debate in literature. People don't know if this carbonate is formed during the sequestering of CO2 with these types of materials. And we can clearly see that, yes, so we see a little shoulder in the dry sample. This is probably because some water got into the system. But in theory, bicarbonates cannot form under completely dry conditions because it needs water to form the bicarbonate species. So this is very good findings into the fundamentals of how CO2 is absorbed, and we hope to publish this soon. So onto the other topic, this is the biodiesel production. So it's a transesterification reaction in which we start from fatty acids, carboxylic acids with long polymer chains. We react that with an alcohol, in this case methanol, to tie in with the previous project, and we obtain an ester, which has uses in the biodiesel industry, so it can be used as a fuel. And for this project, we're using hydrothermal carbonization, which is commonly known as HTC. The HTC uses biomass. So we start from biomass and water, and we put this in a reactor, a sealed reactor called an autoclave. And under fairly mild conditions, we take this sealed reactor into an oven and we heat up to 250, but usually it's more close to 200 degrees. And the water generates pressure within the vessel. And it's this self-generated pressure and high temperatures, well, relatively high temperatures, that produce the formation, the breakdown of the biomass into smaller monomers that then decompose and condense into forming carbonaceous materials. So after an HTC reaction, we have three phases. We have a gas phase that can have CO and CO2 and various volatile compounds. Then we have a liquid phase that's enriched in these decomposition products I mentioned before. And then these decomposition products are reactive and they react with each other, condensing, forming a solid phase, which we call hydrochar. So a very brief overview into what happens inside an autoclave. We can see in this very simple schematics. So we start with saccharides that they first hydrolyze into smaller monomers, which can be sugars, mostly glucose is the most abundant. And then these monomers decompose, dehydrate, and then fragment into smaller decomposition products that are more reactive. And then these products further decompose into aldehydes, phenols, or organic acids that remain in this liquid phase, as I said, or maybe the gas phase if they're volatile. And the rest of the products polymerize and condensate through various complex aromatization reactions and condensation and dehydration. And they form what we know as hydrochar, which is a black material. So using hydrothermal carbonization for producing these carbonaceous materials has advantages and disadvantages, as we know. Everything has. So as a disadvantage, the main disadvantage of these materials is that they lack porosity and surface area. For an application in catalysis, we want the material to have as much surface area as possible, because in catalysis, the contact between the reactants and the active phase of the catalyst it's important. So by increasing the surface area, we're allowing the reaction to take place, more cycles to take place of the reaction. But the advantages of using hydrochar is that we are revalorizing waste biomass, so we can take any waste biological product and use it as a carbon source and produce these materials, these functional materials. And another advantage is that these disadvantages can be remedied through various techniques, such as physical and chemical activation and various techniques that allow the formation of porosity, induced porosity in the materials. So in this project, we used waste milk as the aqueous media in the preparation of hydrochar. The thought of using waste milk is that, at least here in Sweden where I'm living, there's a lot of waste milk, and there's a lot of production of milk, and throughout these production, there's a lot of waste milk that is not up to standard, or that is dirty, or it's not readily available to commercialize. So finding something to do with this waste milk instead of just throwing it away seems like a feasible idea. So we did this HTC with waste milk and biomass. And through XRD, we showed that the materials presented phases of calcium and calcium magnesium phosphates, which are basic sites. So what's interesting about the basic sites is that they give the solid material some sort of basic activity to them. So we know that this transesterification reaction is catalyzed both by acid and by base. So if we can use these basic sites as a catalyst for this reaction, that would be great. And the textural properties of these carbons formed through the HCC of milk and biomass are comparable to standard activated carbons. So using milk does not disturb the porosity and the textural properties of the carbons formed. And as an added bonus, the catalysts seem to be magnetic due to the presence of iron species after the hydrothermal carbonization. So removing them from the reaction media, which is easy just by using a magnet, we can separate the compounds easier. And as to why we want to use milk to produce catalyst to be used in biodiesel production, so we can see in this graph, we have palm oil production, which is one of the oils that can be used for this transesterification reaction. One of the main importers and producers of palm oil is countries in South Asia, mainly Indonesia. So we can see there's a correlation between increase in population and increase of waste production, milk production. So more milk production leads to more milk waste generated. And with them being the main importers of this oil, it's easy to see a relation between having waste milk, converting that into an active catalyst that you can use your own produced palm oil to produce biodiesel. This biodiesel production is especially interesting in developing countries because in highly developed countries, there's an issue between the food versus fuel debate. So a rich country would be spending farm crops and money into producing food that they would not use as food and they would use as fuel. So it's quite strictly regulated how much a country can spend on this feedstock for biodiesel versus using it as food, because it doesn't seem fair that a country that's rich produces food and then uses for fuel, and then there's countries that can't afford either fuel or food. And then for some conclusions, we can say that a supported system has been prepared and proven to work in the hydrogenation of CO2 to methanol. This is a rather new process, so we still need to optimize and tune some parameters for it to be 100% active, hopefully. But we know it works and it's a new system. Then further studies into the CO2 to methanol reaction are underway through various computational experimental NMR experiments. So we'll be able to know how the CO2 is captured by these materials and hopefully enhance these properties. By knowing what sites are more active than others, we can find out a better synthesis procedure to make more active materials. And then activated carbons can and have been prepared from hydrochar, through hydrochar, hydrothermal carbonization using biomass and milk. And the milk gives basic properties to the solids. So the future work on testing the applicability of these materials in this transesterification reaction I mentioned. And then last but not least, this fundamental understanding of the bicarbonate species detected through solid state carbon-13 NMR, both by direct detection and cross-polarization techniques. meaning that we can contribute to literature regarding the study of these techniques. And that's the end of the presentation.